Today we are going to discuss about electron transport chain. Electron transport chain. What is the use of this electron transport chain? It helps in synthesis of ATP. Where do you see this electron transport chain? In which organelle we will see this electron transport chain? In mitochondria. That too especially inner mitochondrial membrane. Okay. So for this electron transport we require four complexes. Okay, for electron transport. Complex 1 and complex 2, they will transfer electrons to ubiquinone. This is Q. This is called as a Q. And complex 3, they will transfer electrons from ubiquinone Q to cytochrome C. And complex 4, from cytochrome C to oxygen. They'll transfer electrons from cytochrome C to oxygen. Now we'll look in detail about each complex. First we'll see complex 1. What happens in the complex 1? This complex 1 is also called as NADH Q oxidoreductase. Okay. Why we are calling this as an NADH Q oxidoreductase? Because from NADH electrons are transported to Q. That's why it got a other name called as NADH Q oxidoreductase. It is in a L shape. In this L shape, this part, vertical part, is projected into the matrix. The L part is present in the inner membrane. Which inner membrane? Inner mitochondrial membrane. Okay. This complex 1 has multiple coenzymes and cofactors. Okay. What is happening here? NADH will be there. Okay, will be produced. From where do you get this NADH? Mostly from citric acid cycle and through fatty acid oxidation, you'll get NADH. This NADH will donate electrons to FMN. Okay, FMN. 2 electrons are donated by NADH. These 2 electrons are transferred to iron sulfur clusters, which are present in the complex 1. Iron sulfur clusters. 1 iron is occupied by 4 sulfur atoms. Cysteine will participate in this iron sulfur cluster. Okay. Cysteine will participate in this iron sulfur clusters. And normal sulfurs also, inorganic sulfurs also participate in iron sulfur cluster. Inorganic sulfur will also participate in formation of iron sulfur clusters. It can be in the form of 2Fe, 2S, 4Fe, 4S, 1Fe, 1S. Okay, these are in the different forms. Okay, from NADH, 2 electrons are transported to FMN, and this FMN is converted into FMN H2, H2, and from this FMnH2, two electrons are transported to iron sulfur cluster. From iron sulfur clusters, two electrons are passed to Q, ubiquinone. This ubiquinone, by taking up of two electrons, it is converted into a free radical form, and this free radical form will accept two hydrogen ions from the matrix, and it will be converted into QH2. Okay, it is a diffusible one. It moves, this ubiquinone moves from one complex to another complex easily. Okay, it can easily diffuse from one complex to another complex. This transfer of electrons from matrix to this towards this intermembrane space or inner mitochondrial membrane, this will create a electro gradient, and this causes movement of 4 H plus ions from matrix. This is matrix side. Okay, 4 H plus ions will move from matrix side to intermembrane space. Okay, 4 H plus ions will move from matrix side to intermembrane space. Okay, this is about complex 1. Next we'll see complex 2. Complex 2. This is called as succinate Q reductase. Succinate Q reductase complex. Succinate Q reductase complex. Okay. For this succinate Q reductase complex, there is a speciality. What is that speciality? This succinate dehydrogenase enzyme is a part of succinate Q reductase complex. Okay. What is the function of this succinate dehydrogenase? Succinate is converted into fumarate. Okay. Succinate is converted into fumarate, and FADH2 is formed. FADH2 is formed. Okay. This FADH2 will transfer its electrons to iron sulfur cluster. From iron sulfur cluster, series of iron sulfur clusters, these electrons are passed to Q, and this Q ubiquinone is converted into QH2. Okay, this complex 2 is very simple. Okay, this FADH2 comes from citric acid cycle. Okay, next complex 3. Next is complex 3. What happens? You can name this complex 3 as Q cytochrome C oxidoreductase. Okay. This complex 3 has two cytochromes. This complex 3 has two cytochromes. Okay. They are named as cytochrome B and C1. Okay, and this complex 3 has 2Fe 2S iron sulfur cluster centers. This is called as Rieske center. What is the speciality of this Rieske center? In complex 1, I told that iron sulfur cluster is occupied. This cysteine is one of the component. Okay, instead of cysteine in this complex 3 iron sulfur cluster, they will have histidine. Okay, instead of two cysteine residues in complex 3, this Rieske center has two histidine residues. Okay, this is the speciality. Okay, and one more speciality of complex 3 is they can accept only one electron. This cytochrome C will accept will accept only one electron. Okay, it will accept only one electron, but QH2 has two electrons, okay. Then there is a difference. Only cytochrome C can accept only one electron, but QH2 will donate two electrons. So this discrepancy has to be cleared. How it is cleared? It is done with the help of Q cycle. Okay, this discrepancy is cleared with the help of Q cycle. Okay, what happens in the Q cycle? For Q cycle to occur, you require two QH2 molecules. What happens in this Q cycle? We require two cytochrome C. Molecules. Okay, this is one thing. Here one cytochrome C is there. Okay, this is cytochrome C, and this is one more cytochrome C. Okay, cytochrome C. Then we have QH2, two QH2s. One electron is donated to cytochrome C1, and again it is transported to cytochrome C. Okay, that is one electron. One electron is transported from QH2 to cytochrome C. One more electron which is present in the Q is transported to Q. Okay, ubiquinone. This is converted into semiquinone. Okay, one free radical, one is called as a semiquinone. Okay, this is called as semiquinone. Where do you get this ubiquinone? We have a pool called as Q pool. This Q pool, from Q pool only we are getting QH2. Okay, and this QH2 will donate one electron to 1Q and one electron to cytochrome C1, and it is transferred to cytochrome C. Okay, from this QH2, two electrons are transferred, and we'll get only free Q. One Q is semiquinone, one is normal quinone, and one electron is transported to cytochrome C. Okay, this semiquinone is formed due to donation of one electron to Q, ubiquinone. Okay, then this Q will join Q pole. From Q pole, again one more QH2 is formed. Same this thing will happen here also. This will donate one electron to cytochrome C1. Okay, and this electron is transported to cytochrome C, and one more electron comes to this part. Okay, semiquinone part. Okay, semiquinone part. Okay, this one. Okay, this semiquinone Q will take up one more electron and one electron. Okay, this will become QH2, and you will get Q here, and they will both participate in a Q pool. Okay, so in complex 3 you require two QH2 molecules and two cytochrome C. Okay, and from matrix, two H plus ions are transported and they will join with Q. Okay, due to this electron gradient, 4 H plus ions are pumped out. Okay, to intermembrane space. Okay, intermembrane space. Okay, in complex 1, we are transporting 4H plus ions out. Okay, 4 protons out, and in complex 3 we are transferring 4H plus ions. Okay, 4H plus ions. But in complex 2, no protons are pumped out. No protons are pumped. Okay, no protons are pumped. So that means one less ATP will be synthesized whenever electrons are transferred from complex 2. If it starts from complex 1, you'll get extra ATP. Okay, extra ATP. We'll see in detail in the next part. Okay, next part. Okay, this is about complex 3. I think you have understood Q cycle. Okay, in complex 3, cytochrome C can accept only one electron, but QH2 has 2 electrons, and this discrepancy is cleared with the help of Q cycle. Okay, in Q cycle, this QH2 will donate 1 electron to cytochrome C and 1 electron to ubiquinone. This ubiquinone is converted into semiquinone, and it will further accept one more electron from another QH2, and it will become normal QH2, and one more electron is donated to cytochrome C. That means in complex 3 we require 2 QH2 molecules and 2 cytochrome C. Okay, next we'll discuss about complex 4. This is cytochrome C oxidase. Okay, in this what will happen, electrons are transferred to oxygen molecule. Okay, for this transport we require 4 cytochrome C. Okay, 4 cytochrome C. We'll see the series of reactions in complex 4. This complex 4 will contain copper containing coenzymes. One is heme A. One more is heme A3, and this one is copper B. Okay, copper B. Two cytochromes will donate 2 electrons, and these electrons will pass from copper A to heme A to heme A3. This will contain iron. Okay, from this they are transferred to copper B, and here what will happen, oxygen molecule should be broken down. Okay, why it has to be broken down? In complex 4 we have to get water molecule. This water molecule will contain only one oxygen atom, and we have to break this oxygen atom into two single atoms. O2 has to be broken into 2 O's. Okay, 2 O's. This is done with the help of passage of electrons from cytochrome C to copper A and heme A3 and copper B. What will happen here? This is copper B, this is heme A and heme A3. Okay, this is copper B. There is a peroxide bridge formation between copper B and heme A3. Peroxide bridge formation will be there. This is called as a peroxide bridge. Okay, after that again 2 cytochromes are utilized. 2 cytochromes. I said 4 cytochrome C are required. Why 4 cytochrome C? Each cytochrome will have only one electron, and 4 cytochrome C means 4 electrons. 4 electrons are required to break the oxygen bond. Okay, and this is cytochrome C. Okay, 2 cytochrome C. Cytochrome C. Okay, from this cytochrome C, again electron passes from copper A to copper A. This and this passes to heme A, and this passes to heme A3, which contains iron, heme A3, and oxygen. Okay, with the passage of this electrons, this copper B, okay, there is a transport of H plus ions from matrix into the complex. What will happen here? Oxygen will bind with the hydrogen atoms, and they will form OH, hydroxyl groups. Okay, next, this is again what will happen here. Again they will take two hydrogen atoms. Okay, two hydrogen atoms. What they'll do, again this is normalized into, again it will participate in the cycle. Okay, this goes back here, and with this we'll get two water molecules. Okay, two water molecules. We have two oxygen atoms. Okay, one oxygen atom. Okay, O2. O2 is converted into single O and single O, and we are getting H and H attached in this part. O is attached with H and H. Okay, and one again two hydrogen atoms are taken up, and you are getting H2O and H2O. That means two water molecules are getting out. Okay, getting out. In this pathway, 4 H plus ions are pumped into intermembrane space. Okay, from complex 1, 4 H plus ions. Complex 2, no H plus. Complex 3, 4 H plus. Okay, here I'll tell you what happens in the complex 4. Okay, complex 4. Here, how many electrons we are utilizing in complex 4? We are utilizing 4 electrons. Okay, 4 electrons. But in complex 1, how many electrons we are utilizing? 2 electrons. Here also 2 electrons. Why 4 electrons are required in complex 4? To break oxygen, you require 4 electrons. Okay, so the reaction is starting with 2 electrons. For each 2 electrons, you are getting 4 H plus. Okay. Okay, then here you are utilizing 4 electrons. If you take 2 electrons, how many H plus ions will come out? Only 2. If you take 2 electrons, then only 2 H plus ions will pumped into intermembrane space. Okay, here I have written 4 H plus ions are pumped into intermembrane space. But for how many electrons these 4 H plus ions? For 4 electrons, you are getting 4 H plus ions. For 2 electrons, you will get only 2 H plus. Okay, then reaction will be like this. Instead of 2 H2O, you have to write 1 H2O, half oxygen. Okay, you'll write in the reaction as half oxygen. Okay, half oxygen, and you'll get 1 water molecule and only 2 H plus ions. At last, what is this? Complex 1, 4 H plus. complex 2 0, complex 3 4 H plus, and complex 4 only 2H plus. Okay, 2H plus. For 4 electrons in complex 4, you are pumping out 4 H plus ions. For 2 electrons, there is only pumping of 2 H plus ions. 2 H plus ions. If you see the reaction, only one water molecule is generated for 2 electrons. Only one water molecule will be generated for 2 electrons. In complex 4, you are utilizing 4 electrons. Why you are utilizing 4 electrons in complex 4? If you are not using 4 electrons, if you break this oxygen, free radicals will be generated. With 2 electrons, you can't break this bond perfectly, and there will be generation of free radicals. In order to prevent that, you will require 4 electrons. Okay, that's why you are using 4 cytochrome C. But the reaction is starting with 2 electrons, and ultimately if you take 2 electrons, you will have 2 H plus ions, which are pumped out from complex 4. Okay, complex 1 is 4, complex 3 is 4, and complex 4 is only 2 protons are pumped out. Okay, complex 1 and complex 3 4 protons, and complex 4 is only 2 protons. This ends with the part 1 of electron transport chain. Okay, thank you.